Well, welcome everybody. My name's Keith Burnett. I'm the Vice Chancellor of the University, and it's really a delight and honour to welcome you to this evening to hear Sir Paul Nurse speak to us. Sir Paul is an extraordinary scientist, President of the Royal Society since 2010, a Nobel laureate. In fact, the first time I heard you speak, Paul, was at the Nobel ceremony in 2001, and you've done so many extraordinary things. He's a geneticist, cell biologist, and his discoveries have helped explain how the cell controls its cycle of growth and division, but all aspects of work which has broad significance, and that's why, of course, his work was given the Nobel Prize for it. It has impact not just in what some people might think of as narrow domain of biology. We don't think like that. This is the KrebsFest, so biology is everything. But it's got to do with cell growth, division, how they cause cancer and serious diseases. Extraordinary. So, in fact, Paul's contribution to cell biology and cancer research led to a knighthood in 1999, and as I said, in 2001, the Nobel Prize for Physiology and Medicine. But also you've played very, very many senior roles, both in universities and in science. In 2010, you were elected to the presidency of the Royal Society, and in 2001, you became the Director and Chief Executive of the Francis Crick Institute. I just want to say really that these are extraordinarily important roles in British science and in society, and you've fulfilled those roles in an extraordinary way. This was a very important day for some of you know. There was the spending statement from the Chancellor of the Exchequer in Parliament, and I know really the effort that Paul has put in actually explaining the value of basic science to our political masters, explaining why it needs to be supported in the way, explaining the importance of biology in its broader sense. And I just want to thank you really for that work put in, Paul, because it's seen in the statement that the way you've fulfilled that has been very, very important to our future. So thank you for that. So Paul's subject this evening is the great ideas of biology, and it's part of our Krebs Festival, which celebrates the work of Sir Hans Krebs, a Nobel laureate himself, but it is a real pleasure to welcome you to celebrate with us the tradition that Hans Krebs both developed and the tradition that carries on in Sheffield to this day. It's wonderful to welcome you to give your talk about the great ideas of biology. So everybody, let's help me welcome Sir Paul Nurse to give us this KrebsFest lecture this evening.
Well, thank you very much, Keith. That's a very nice introduction. Great to be here in Sheffield. Great to see you all here for this lecture. It's actually a real honour and pleasure to make a contribution to the KrebsFest because, of course, Krebs was one of the great biochemists of the 20th century. And he spent significant time here. He was a demonstrator, then a lecturer, then professor at Sheffield, and head of the Department of Biochemistry. He then subsequently went to Oxford, up or down, we'll leave that for you to depends on which way the trains run, perhaps, where he was the Whitley Chair of Biochemistry, and I was also a professor in the Biochemistry Department in the 1980s, so I had that connection too. His great contribution, of course, was in metabolism, and his discoveries of the citric acid cycle and the glyoxylate cycle some years later. And the citric acid cycle, as I'm sure many of you know, is, of course, known as the Krebs cycle. And the point I want to mention tonight is that is common to all life. And that influenced the choice of the talk that I'm going to give tonight, because it was this universality of the Krebs cycle, this underpinning of the metabolic processes of all living organisms, which led me to choose the topic today in honor of Hans Krebs, on the great ideas of biology, where I've tried to focus on universals that underpin the nature of life. And as such, it's written really in respect of Hans Krebs. So that's why I'm really pleased to be here. So I'm going to talk about great ideas of biology. I have two sort of affiliations, I should just mention. I'm president of the Royal Society for, I think, another 4 days. So I'll have to change this next week. But I'm also leading up the Francis Crick Institute. And I mention this because should you ever go to London on that nice train, and should you look out to the right as you get off it, you will see about 10 meters away the front of this building. And it's just been completed, and it will have about 1,600 scientists in it when it's completely full. Now, I'm going to talk about the great ideas of biology. Now, on the whole, biologists tend not to talk much about great ideas and grand theories. In this respect, they're rather different to physicists who love doing that sort of thing. And us biologists tend to be rather more comfortable with details and particulars. You know, the sort of things we would do is count the numbers of species in a habitat. Or if we were a 19th century biologist like Darwin, we'd count the numbers of hairs on a beetle's leg. Or today we sequence genes, we determine the RNAs and proteins in a cell. It's all much the same, lots of detail. But the truth is, biology does have some great ideas. And I want to talk about 4 of these tonight, which I think are not very controversial, and point in the direction of a 5th, which I think is yet to be fully formulated, but which I think is also important for understanding life. Because all of these ideas contribute to understanding the nature of life. All of them have universal characteristics reflecting Krebs and his cycle. I'm going to start with the idea of the cell. And in this talk, I'm going to give something about the history of the ideas, something about the philosophy behind it, and I'm going to emphasize connections between the different ideas. So this isn't going to run through everything that was done in the last 3 years. I'll be extending back over 3 centuries. Now, the idea of the cell, and how would one define it, is simply stated. All life is composed of cells, and the cell is the simplest unit exhibiting the characteristics of life. 2 ideas in that. One is that the cell is the basic structural unit of all life. If you like, it's life's atom. And secondly, it's the basic functional unit of life. That is the simplest entity which has the characteristics of life as we'd understand it. So it's a critical idea to biology. And it's an idea that was started in the 17th century, and it was started with an early fellow of the Royal Society, Robert Hooke, and he published in 1665, using a microscope you see there on the left-hand side of the slide, he published that black and white picture in Micrographia, and it's a picture of a slice of cork from a plant taken with a thin razor and then looked at under the microscope. The microscope had been invented in Holland about 40 years previously, but hadn't been used in this fine detail in looking at living things. By the way, on the right is a modern scanning electron micrograph of the same sorts of material. When he looked, and he was working in Oxford at the time, and there's a plaque on the High Street in Oxford that records the discovery of the cell. When he looked down at the microscope, he saw these pictures that you see here. They reminded him of monk cells, of a plan of a monastery, and so he called them cells. And that was the origin of the word cell. Now, this is an example of technology begatting discovery. In this instance, it was the technology of the microscope. But a second feature, which is often common in science, also followed, because once one person has seen something in science, everybody sees it. Once it's been discovered, it spreads like a virus. And within 1 or 2 decades, cells were discovered everywhere, and sometimes the illustrations were of extraordinary quality. This is actually a picture, the one on the left here, from a book I own from 1682 by Nehemiah Grew, also a fellow of the Royal Society. He was a doctor. And this is a section of a vine plant, and it's of a quality that you would have found in a mid-20th century textbook on plant anatomy. On the right, you'll see another scanning EM, just to show what it looked like. It was easy to see cells in plants. They have thick cell walls, no problems with fixation and the microscopy. More difficult to see them in animals. The first animal cells were from blood, and these were described by Malpighi and by Leeuwenhoek. And Leeuwenhoek also discovered the first single-celled life, microorganisms. This is a picture that is in the ownership of the Royal Society. He sent all his observations in 1,000 plus letters to the Royal Society during the second half of the 17th century. And this is particularly charming. What he did is he scraped between his teeth, which he used to clean regularly and was rather shocked by what he found when he looked under the microscope. And he saw all these bacteria. And you'll see there under figure B, a little bacteria doing a loop-the-loop, which I imagine it was still living and swimming around under the microscope. This was all done with a single lensed microscope, which it reduces chromatic aberration, so it's got good definition, but extremely difficult to actually make and use. Now, Leeuwenhoek was actually a draper. He wasn't a scientist. I mean, they didn't really quite exist properly then. He was an artisan, and he lived in Delft. And it turns out that he was the executor of the will of the painter Vermeer, who also lived in Delft at around the same time. Now, Vermeer, unusually, he doesn't have a big output, about 40 paintings. He unusually did a painting of the same man, 2 paintings, this is the one from the Louvre, of a scientist. One's called an astronomer, the other is called the geographer. And there is an idea around that maybe he had painted Leeuwenhoek, because obviously they knew each other. And I've just read a recent theory that Vermeer may have carried out many of the original drawings that Leeuwenhoek made under the microscope. He discovered them, sketched them, and then had a professional to actually finish them. And what's interesting is that the style of those illustrations changed the year that Vermeer died. Now, I'm encouraging this idea because we have 1,000 of Leeuwenhoek's illustrations, of which around 500, you never know, might be Vermeer's. And so I'm thinking of increasing the insurance of what we have there. Now, that was up to about 1700. Over the next 100 years, and it just stated in a slow way, with better microscopes, better histology, better fixation, better techniques, gradually cells were discovered essentially everywhere, in animals and all plants, and they seem to be assemblages of what Leeuwenhoek had called animalcules, that's these small single-celled organisms. And by 1839, the cell doctrine was born, and it was defined by 2 German scientists, Schleiden and Schwann. Here's Schwann, rather serious-looking gentleman, I always think. And this is from his book in 1839, translated from the German. We have seen that all organisms are composed of essentially like parts, namely of cells. So this is the basic structural unit idea that I mentioned. A few years later, Rudolf Virchow, who, another rather serious-looking German, but who was in the government of Bismarck, actually. He was the founder, really, of pathology. He emphasized the functional significance of the cell with this sentence from 1858. Every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life. So that is the simplest entity that defines what life is. This is a major milestone in biology. It's not quite recognized as such in some respects, and some of the other ideas will be more familiar to you. I mean, this is familiar, but it was an extremely important idea. The cell underpins all reproduction and all development. It has extraordinary complex structure. Here's a modern EM. Here is a mammalian embryo, and if you can just see the dots there, you probably can. Each of those is an individual cell. So we cannot understand reproduction, we cannot understand development, we cannot understand diseases like cancer unless we understand what the cell is and how it works. It's absolutely crucial. And should I have failed to raise your interest in cells, I'm going to show you one more picture. This is an egg being fertilized by sperm. And I'll remind all of you in this room that you were all once, just like this, you were all once a single cell. And if I fail to excite you, there's a door at the back, because it goes downhill after this. So the cell continues to be important. It's the basic unit of life, important for reproduction and development. And we can read about stem cells, for example, almost daily in the newspapers. It is still a major topic of contemporary interest to us scientists, but also to society as a whole. My second idea, the gene, basis of heredity. Now, this is perhaps more familiar, the science of genetics, more familiar as an important subject from ancient times. I'm talking about the classical Greeks. It was observed that particular characteristics that an individual might display appeared to be passed down from generation to generation, from parents to offspring. And speculation was made, what could be the basis of this heredity? And this was a problem that was being tackled in the middle of the 19th century by plant breeders who were interested in improving crops, fundamentally, and were crossing plants with different characteristics to see how those characteristics were inherited from generation to generation. And it turned out that it was rather complicated. The characters sometimes became blended in the offspring, something that was intermediate. Sometimes one of the characters would disappear and then reappear in subsequent generations. And it was very difficult to make much sense of this from the biologists who were studying this problem. That is, until Gregor Mendel came along. He was I think of him as a gardening monk. He was a monk in Brno in the Czech Republic, then in the Austro-Hungarian Empire. And he belonged to an order of monks, a monastery, of essentially scientific monks. It's quite a difficult concept for us today, but that particular order was involved in teaching in the gymnasia, in the senior schools, and also did research. Mendel was taken in as a young man. He was trained as a physicist. He didn't do very well at examinations and never got qualified to formally teach physics. He didn't pass his examinations in the University of Vienna. But he turned his mind to both meteorological problems. Actually, he studied the drop in barometric pressure during a storm, for example, but also to plant breeding. And he argued that to sort out what was happening in these complex crosses, you needed to take a rather fresh approach to it. And these, I think, derived from his physical training, because he thought biologists, naturalists tend to go to description, as I've already implied. Physicists tend to be more quantitative and also more abstract in their thinking, and also to focus more on theories. And what Mendel argued was that you needed to both accurately describe the different types of what we would call phenotypes, that is, the characteristics of the plants that were appearing. You needed to describe those very clearly, and not only describe them, so catalogue them properly, but also count them. And mostly that didn't happen, though I'll have an exception in a moment. And then to try and think about what it meant. And I visited, actually, his monastery for the first time in 1981, actually, in the middle of the Cold War. And he had a major research activity. This is his garden. This is a photograph I took, actually, in the 1980s. And that whole garden there was used for his plant crosses. Next to it was a greenhouse of similar size, which was built for him so he could carry out his crossing experiments. I mean, the research councils would be jealous of what was being put in place here. And this it was in the 1850s and 1860s. Now, he was successful. So what were the reasons for his success? Well, I'm not sure. I'm not a Mendel expert. Actually, I'm not an expert on anything I'm going to talk about tonight, so do take it with a pinch of salt. But I think there's three reasons for his success. One is he chose material that he could understand. Now, that sounds a little bit like cheating, okay? What he did is he did trial experiments with 6 or 7 different species, and most of them he couldn't make any sense of. But with the pea, and that's an example of the pea that he worked on, the crosses led to simpler patterns of inheritance. And the reason I emphasize, the reason is actually that fewer genes were involved in the traits that he was looking at. If you have many genes involved, it just gets too complicated. And I mention that because it's very important in biology, actually in science in general, but particularly in biology, to choose material, to choose topics and, that you can study in systems where you have a chance of making sense of them. It's no good having a grand problem and a grand ideas if you can't make any sense of it. And choosing the right material and choosing the right problem is really critical. He chose this material partly because there were variants in the peas that were very clearly distinguishable. That is, plants of different heights, for example, different coloured flowers, which tended to be complex, but also different seeds and colours and shapes and so on. Second thing that I think was important, I've already mentioned it, is he recorded this accurately and counted what he saw. The third is that he generated a theory or a law to explain his observations and then went about testing it. Now, this is more or less how we do most of our science today. It wasn't how the naturalists behaved generally in the 19th century, so this was different for biologists. What did he observe? He observed that in the first generation after a cross, one character, if you were crossing a tall or a short plant, for example, one character often dominated in the first generation, and one disappeared. I'll show you here, this was to do with the colour of seeds, and you see a green cross to a yellow. The yellow dominates in the first generation. But then that original character, in this case green, coloured seeds, would re-emerge in the second generation, as you see here. The important point was, if you counted them, they emerged in distinct ratios. This is the famous 3-to-1 ratio that many of us would have learnt about in school. And what that seemed to him, because he didn't just describe it, he then thought, what could explain it? And perhaps his more mathematical leanings from his physics training made him think that this looked like combinations of particles in some way that were sorting out. Now, one thing I discovered literally only a couple of years ago is that Charles Darwin, and I'm going to speak more of him in a moment, Charles Darwin also did crosses in plants. He did it in snapdragons, and he published within 3 years of this, not knowing about Mendel's work, a 3-to-1 ratio in snapdragons. Exactly, not the same numbers. Mendel counted 3 or 4,000. He only counted 100. But he recorded 3-to-1 and just noted it and then moved on. What Mendel did was to try and explain it, and he explained it in a sort of combinatorial theory. So his observations, and I'm not going to go through Mendelian genetics, but it led him to propose what is essentially a particulate theory of heredity. And that is described as follows, that there are particles, we'd now call them genes, that determine character differences in the plants. That in any particular plant, there are 2 particles that are important for a character difference when you're looking at different phenotypes. But only one of those particles passed into each gamete that's formed when the plants reproduce. However, on fertilization, that is when the pollen fertilizes an ovule, each would bring one of those particles, so you restore the duality, the 2 particles. And finally, he proposed that the characteristics of one of those particles could dominate over the other in determining what the character looked like. And by applying those ideas, which were a brilliant piece of abstract reasoning, he could explain all the numbers and everything that he did. What happened was he published 2 papers in 1865. Everything he wrote there was completely right, and not one person took the slightest bit of notice of it. Not one person. It wasn't that it was published in obscure journals in Middle Europe. He sent them to a variety of people. It's mentioned, I've discovered, even in the Encyclopedia Britannica of the 1880s. So it was, but not the idea, just the observations. So it wasn't recognized for what it was. Then, at the beginning of the 20th century, 3 groups reproduced his results and confirmed that he was right. The first group that published it actually ignored Mendel. The second group, having been scooped by the first, obviously went through the literature to see if anything had been published before, and delighted in saying it's already been done in 1865, 40 years earlier. Nothing much changes in the personality and character of scientists or academics, I have to say. So why was it that this was accepted in 1903, 1905, and not in 1865? I don't, of course, know, but I do know that certain things were discovered in those 30, 40 years that could have been relevant. The main one was the discovery of chromosomes. This is a late 19th century slice of an onion root, which many of us would have looked at school. And what you see here are these threads, which are the chromosomes separating every time a cell divides. And when these were observed, it was thought maybe these chromosomes had the determinants of heredity, since they seem to equally partition components from one cell to another. And critically, a scientist called Van Beneden, working again on suitable material, which in this case was the nematode worm, which only had 4 chromosomes, unlike the onion, which has got many. Then when Beneden counted the chromosomes, what did he note? When the worm reproduced and made gametes, they only had 2 chromosomes. So now you'll recall in Mendel's theory you had reduction in half in the numbers, and that fitted with the abstract reasoning of Mendel. So what we have here is really abstract reasoning that now becomes supported by concrete observation. And that makes a much more comfortable sort of feeling. It's not just an abstraction, it also has clear data. So this was a major advance, and much of the important discoveries in biology in the 20th century were turning this idea of Mendel's into a molecular understanding, starting with the discovery that DNA was a hereditary material made by the ability to transform pneumococcus so that with deoxyribonucleic acid. That was done in Rockefeller University in 1944, where I was president for 7 or 8 years. And it was done by 3 researchers, Avery, MacLeod and McCarthy, whose work I'd read about when I was a student in the 1960s. When I went to Rockefeller in 2003, I think it was, they gave me a cocktail party to welcome me, and this little old man came up to me to shake my hand and said his name was Mac McCarthy. Now, I, of course, thought that McCarthy had died already by the time I had taught in 1968. So I just looked at him and I said, "You can't possibly be the Mac McCarthy." And he said, "Yes, I am the Mac McCarthy." And he was 92 at the time. He did die 3 years later, and was still there working at Rockefeller University. He was one of the three who discovered what's called the transforming principle, which is DNA. And that showed the chemical basis. And we know Watson, Crick, Franklin, Wilkins got the structure of that, and this I'll talk a little bit more later. The ladder and double helix of DNA, and then Francis Crick introduced what's called the central dogma, where the DNA encodes RNA, that's the transcription, that's then translated into protein, the proteins fold up and determine characteristics of living things, and that you can then refer back to the gene, giving the linkage that Mendel was noting between the particles, the genes, and the subsequent phenotype of the plant. So that's my second idea, the gene, basis of heredity, gives you an explanation from the genotype, the genetic composition, to the phenotype, what the organism looks like, and is always eternally interesting for us as human beings. Just one thing there. What does it mean to be human if our phenotype, our characteristics, has a significant determination by genes? And the interaction between genes and environment, extremely interesting. What does it mean for crime and punishment? You know, if one is genetically determined. These are complicated issues that we need to take care of. Third idea. Well, it has to be evolution. If you go into a popular bookshop, Waterstones, I'm sure we have Waterstones in Sheffield, and you looked at the popular science shelves, you could be forgiven for thinking there's only two topics in science. One of them is evolution, and the second is string theory, because there's endless books about both of these topics. Well, this one, I'm not going to talk about string theory. You'll be relieved to hear. And actually, I'm relieved as well. That's the sort of thing my daughter works on as it happens. But I'm going to talk about evolution by natural selection. Within this, there are two components to this idea. The first is that life evolves. That is, there is a continuity between all organisms on the planet. And the second is, a major mechanism is natural selection. And I'm going to consider both of these components of these ideas. Now, they're both usually associated with Charles Darwin. That's the man there on the right, when he was a sort of youngish man in his late 40s, early 50s. But the one I'd like to talk about first, because is not him, because the idea that life evolves was not Charles Darwin's idea. It had been around, some would say, even since Aristotle, but certainly for 100 years prior to Darwin. And one of the proponents was Darwin's grandfather. And Darwin's grandfather is on the left there, and he was Erasmus Darwin. And he was a really fascinating, eccentric character. He was a doctor who lived in the Midlands. He lived in Lichfield, in Cathedral Close, and also in Derby, where you, of course, come through on the train from London to, at least I did, I think that's the only way you can come these days, up to Sheffield. He was a founder member of the Lunar Society. The Lunar Society was this society of academics, though not really much from universities, but thinkers, industrialists, entrepreneurs, and so on, that used to meet once a month under the full moon, so they could ride back to their respective homes And that was they met in Lichfield, they met in Birmingham, for example. You could go to the Soho Works, you can actually go to the room where they had dinner. I always think that they had to ride home. I'm sure they were somewhat the worse for drink when they were going home, so they had to have the full moon to have any chance of actually getting home after that. Erasmus was an interesting man. You can see here, he was fat and completely charming. He cut out his dining room table so he could it could accommodate his stomach, and he could eat nearer his food. He was a charming conversationalist despite having an appalling stutter. He managed to marry the most eligible widow in Lichfield. He was a doctor. Doctors in the 18th century didn't actually cure many people, but he had one extremely good quality, which is that he could tell you when you were very likely to die. And this is an extremely useful thing to know. It means you can make rest with your maker, and you can also deal with that difficult issue of the will and the family without having to suffer for years, rather only days. And so he was usefully charged according to his patient's income. He was a sort of proto-socialist. He got all his income from his rich patients, and the Dean of Lichfield Cathedral didn't like the motto on his coach, which was all things from shells. He was trying to state that evolution, you know, if you open a shell, it's a mollusk, it's a formless mass, and he was trying to argue that all complex life came from these simple forms. And he had to paint it out because he was losing all his respectable patients by having these heretical views. He was asked several times to become George III's physician, which he refused because he had Republican tendencies. He was a doctor, a scientist, and a poet, and wrote most of his science up in verse. And he's such a fascinating... I've actually bought several of his original books in verse. He was a proponent of female education and set up a school for girls. And the teachers were his illegitimate daughters. He was radical in a variety of different sort of ways. Anyway, he argued that life evolved, and he encapsulated it in a number of his poems, one of them called Zoonomia. And it's actually extremely... I'm thinking I have to write a paper one day, a scientific paper in blank verse. I think it would enhance it, I have to say. However, despite having that idea, and he wasn't the only one, Buffon did, and it's particularly a French scientists were important here, it was his grandson, Charles, the man there on the right, who gathered the vast amounts of data that supported this idea of evolution based on both the fossil record and on his observations of living plants and animals. What was important for him was his 5-year trip on the Beagle. He then retreated to Down village on the outskirts of London in Kent, and he eventually wrote his seminal book, The Origin of Species, in 1859, just about the time of Mendel, in fact. And in this book, he had only one illustration. It's here. He drew a tree of life. And, of course, that showed that all animals and plants, all living organisms, were related by descent. I mean, that's what the key issue about a tree of life. Do notice that at the bottom of the tree of life, he has about 8 different origins. He's obviously not clear whether it came from a single life form or whether there were more life forms that were made. And that is quite interesting. He observed that living organisms were adapted to their lifestyles and environment. That was a key point. And this came initially from his study of finches, birds on the Galapagos Islands, which are recent volcanic islands, where there had been diversification from a single finch. He analysed the beaks, which are on the left. I've put on here a sort of variant of pincers, which are used for different things. And of course, the beak can be used for different things, for catching insects or cracking nuts, just as you have different tools for particular different purposes. And from these sorts of observations, he proposed natural selection, which I shall tell you about in a moment, as a mechanism for evolution. And he was stimulated to publish because he received a letter in 1858 from Alfred Wallace, who, and in that letter he described, Wallace did, exactly the same idea. Wallace was suffering from malarial fever in somewhere like Borneo and had written this 30-page sort of letter to Darwin, partly under hallucination, they think, with the same idea. So they published 2 papers in the Linnaean Society in Burlington House, where the Royal Academy is now, and in 1858. Neither of them were there. Wallace was still recovering from malaria, whilst Darwin was rather nervous at public speaking and sent somebody else, it might have been Hooker, the Kew director, to do that. Interestingly, the idea of natural selection was not, although clearly associated with Darwin, had been published previously and unnoticed by a tree grower called Patrick Matthew, who wrote a book on navel timber in 1831, nearly 30 years previously. And there was this rather polite exchange of letters where Matthew wrote to Darwin saying, very interesting book, but you might be interested in my book. And there was a page or 2 in the appendix on tree growing, which is a good summary of natural selection. And you'll see in the first edition he's not mentioned. In the second edition of The Origin of Species, he acknowledges Matthew. Very polite exchange, and Matthew didn't seem to mind that he didn't get the credit. Neither did Wallace. And that's because I think Darwin assembled such a huge set of data and arguments to support his argument. So what is the idea of natural selection? It is that within a species there are variants. These variants are a consequence of inheritable differences. That's the genes I've already spoken about. And those variants which are most successful reproduce more and so pass more of their genes on to the next generation. That's the so-called survival of the fittest, not actually coined by Darwin. It's coined by Herbert Spencer, the sociologist. Therefore, these changes in the gene proportions in a population gradually lead to changes in the characteristics of the species, and that leads to evolution. Now, that's a potted sort of a description of it. Now, I want to repeat this argument, though, and then link it to the 2 earlier ideas, because all of these 3 ideas are actually linked. It's not normally presented that way. And it's based on an argument made by Muller that living things, defining life as living things that have properties which allow them to undergo natural selection and therefore to evolve. It's a sort of a definition of life. And the properties are the following. The properties are that life must reproduce, that life has a hereditary system, and that this hereditary system exhibits variability. And this is, of course, what you can see in a cell combined with the concept of the gene. This is the reproduction of a cell, because evolution by natural selection will emerge from those 2 ideas. Here we have a cell. It's got genes in there determining the properties. Those genes are copied when the cells reproduce. The genes define the characteristics of the cell, and the genes exhibit variability when they're copied or whilst they're being maintained. If you get a mutation, an alteration, then a cell which is, for example, orange, when it divides, could have an alteration in the gene which ends up with it being red. If for some reason red cells are more advantageous and more able to divide more rapidly than orange cells, then that will take over the population. The point I'm trying to make is, once you have the concept of a cell, once you have the concept of a gene, you have, as a consequence of that, the concept of natural selection and therefore evolution. So it's a triad of all these things, 3 things connected together. Well, this is Charles looking a bit older. It's a year before he died. He has these wonderful last sentences in The Origin. Whilst this plant has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Notice the link. This was a biological law that he linked with Newton's law and at the same level. That's deliberate, this connection, to elevate this particular idea. Evolution by natural selection, very important. Probably the most beautiful idea, really, in biology. All life evolves, it's related by descent. Natural selection, major mechanism, not the only one. And then a couple of ideas, contemporary ideas, or more contemporary. Kin selection is relevant to thinking about altruism and morality. And that's something we think about. In the end, science begins to get into ethics and morality. It sort of has this habit of creeping into places where you don't expect it. And the one thing I heard from Guido Pontecorvo, a fungal geneticist, he made the point, the tree of life means we are related to every living thing on the planet. And therefore, it's an argument for us being stewards for our simpler relatives. An argument for the biosphere. A very nice argument that Pontecorvo made. What's my fourth idea? Well, that's appropriate to Krebs. Life as chemistry. That is essentially chemistry, and I won't talk about physics, but physics too, are responsible for the mechanistic basis of life. And that's going to be my fourth idea. This has its origins with Lavoisier, I believe. This is actually one of the great pictures of a scientist that you can find. It's in the Metropolitan Museum of Art. It used to hang in the president of Rockefeller University's office. And then they had one of their financial crises and had to sell it, I think for about $100 million. Anyway, he's got this wonderful leg. I don't know if you can see it there. And that is his beautiful assistant, of course, who happens to be his wife. Now, Aristotle argued that living things had a sort of natural end, a goal or a movement, driven by what he termed an inner vital force. That was the origins of vitalism. And this idea was replaced that you could actually understand life in terms of chemistry and physics. And that was a crucial one. It's my fourth idea. Why do I link it with Lavoisier? Because Lavoisier was thinking about life and thinking about chemistry. And he made comparisons between a guinea pig respiring and the slow burning of charcoal, and did calculations about the CO2 changes and the amount of heat that was produced, and he began to see that there were similarities there. And so he began to think that chemical changes were crucial to the functioning of life. The problem was that he was also a tax collector and an aristocrat at the wrong time in France. And here's another picture of Lavoisier, and you'll see a guillotine in the background because he lost his head. This was a shame, actually, because he was, in fact, in favour of many of the principles behind the revolution. And this is something for us as scientists and academics, to remember when we get too close to politicians. The judge argued, when he was being tried, that the Republic, the French Republic, has no need of chemists or savants. So he lost his head. But he sowed a seed of an idea that was picked up by another great French scientist, Louis Pasteur, in the 1850s, who demonstrated that living microbes were responsible for chemical transformations during the fermentation process. He was working in Lille in 1856 on Eurostar. Seemed to be obsessed with trains, actually, in this talk. Anyway, you pass it on Eurostar to Paris and also to Brussels. And he was approached by the beet sugar fermenters who were making sugar, because sometimes, who were making alcohol from the sugar of beet, because sometimes the fermentations went wrong. And instead of making sugar, they made acid, lactic acid. And what he did was he looked at the fermentation and he saw when it was good and made alcohol, there was yeast there. And when it was bad and made acid, there was bacteria there. And he came to the conclusion that you needed to have yeast for the proper fermentation. So he advised, this is applied science with a truly basic a discovery contribution. He said, add yeast and it'll always work. Okay? So they were very happy about that. He then thought about it and said, fermentation was a physiological act yielding chemical products necessary for the cell. And therefore, such chemical reactions are an expression of the life of the cell. So he took that observation and turned it into a grand theory that is absolutely right, which is you can understand life in terms of chemistry. And that had its origins really with Pasteur. Now, I'm getting a little late here, so I'll go over a little bit here rather more rapidly. We now know that, of course, there is biochemistry. We have enzymes. Enzymes are made of proteins with particular structures that cause changes in between different small molecular weight molecules. Here is a metabolism map, and I put it up here because in red you will see the Krebs cycle. That circle is what Krebs discovered. But the key point here, which I think is really quite interesting and isn't emphasized, is that the chemistry that is going on inside cells is extraordinary. Because in these small objects, just micrometers across, there are hundreds and often thousands of different chemical reactions going on simultaneously in this very gentle conditions of low temperature and these enzyme catalysts. And all this extraordinary diversity of chemistry is going on in the same place. One way I like to sort of rethink about the structure of a cell, which we normally think of, this is a scheme of a cell with a nucleus, Golgi, and so on. It's normally just described as a sort of morphological object. I prefer to think of it actually as a myriad of different chemical microenvironments, where all of these reactions are going on in different environments. They may be in a membrane-bounded organelle, such as the mitochondrion, or it could be in smaller compartments as defined by proteins that give you a small pocket. But the key point is that all of these are occurring in the same place at the same time. The fact that they're distinct and different mean they have to be regulated. That brings something else on top of it, because they have to be connected. You have to keep the chemical environments separate, but they have to be connected. I don't think we sort of think about it like that usually, but that's how I see the structure of a cell. So this is my fourth idea. Chemistry provides a good mechanistic explanation of life. It's the basis, of course, for understanding diseases, which are essentially, we transform disease into chemical explanations, and then the basis of much of the pharmaceutical industry is to look at ways in which you can chemically intervene to restore, change metabolism so you can correct for those particular defects. So an extremely important idea for biomedicine. So this then leads me to my fifth idea, which is still perhaps rather provisional, but it's one that I like, and that is that we can understand life in terms of information. And information is central to explanations in life, and that's going to be my final idea. And really the starting point of that is that most of the time explanations of biology get reduced really to terms that are dealing with the management and processing of information. It's not how we sort of think about it, but I'll give you a couple of examples to try and illustrate it. This management of information is occurring in a complex system of life, and when you're thinking about complex systems, you can get a better insight into life. Now, this idea I thought was quite modern, but I did a bit of sort of digging into it, and I discover a really interesting 25 pages from Immanuel Kant. Kant, K-A-N-T, philosopher of the beginning of the 19th century, on systems biology. It's completely extraordinary, who describes living things in terms of complex systems. But many of those ideas came into sharp focus in the second half of the last century, particularly as information sciences were developed, and particularly as computing, of course, went hand in hand with it. Now, as I've already implied, many biological phenomena can be described in terms of interacting components in a complex system. And a powerful way of making sense of those is to consider the information flow through those complex systems. And this can operate at different levels in biology. Chemical systems in the cells, which I've been talking about and I'll continue with, neuron systems for brains, ecological processes for ecosystems, all can be described in terms of information flow. So the idea is to explain higher order biological phenomena such as homeostasis, communication, reproduction, spatial order, temporal order, all in terms of information. And that's a sort of an objective here. What this approach does is emphasizes regulation, networks, organization, emergent behaviors, all phenomena that are part of information. And in short, the concept is to translate the descriptions of the chemical and physical processes being carried out in cells, and I'm going to focus on cells, into the underlying system and the management of information within that system, i.e. the gathering of information, the storing of information, the processing of information, and how that determines particular outputs. So that's the objective. That's the paradigm that this idea is around. I'm going to give you two examples you'll be familiar with to illustrate what I mean. The first is the double helix. Now, the double helix is the sort of iconic image of biology. And we tend to think about it as just a sort of rather beautiful structure. But actually, the true beauty of the structure of DNA comes when you recognize that actually what it is a digital information storage device. It's not actually the shape of it, it's the fact that it stores digital information very effectively, which is stored in the sequence of the nucleotides that make up each of those strands of DNA. And of course, you can copy it very easily because of the complementarity between the bases that make up those structures. The point I'm trying to make here is that you can describe the structure of DNA in terms of the angstroms and what atoms are touching what atoms, and you get no biological comprehension out of that. The biological comprehension comes when you realize that these structures will store information, and that these structures can easily copy information, giving you the basis of heredity. So that's why I'm putting the emphasis of information here. A second idea. The basis for this is understanding negative feedback control. This is a governor from a steam engine I discovered in New Zealand on a boat that I was traveling on, built in the 1890s. Now, I don't know, you're probably a bit like me, that more or less I can understand most machines built before 1900, and increasingly less and less machines built after 1900. So I have to go for my metaphors back to pre-1900. This is a governor. The steam engine operates, it spins that spindle, those balls are thrown out, it lifts a valve, it cuts off steam going into the engine, and that slows it down. And therefore, you go through an oscillation that governs the speed of the steam engine. Okay? This is a negative feedback control, and we see it here in a metabolic pathway in this case, where a chemical A is changed to B, is changed to C, and C negatively feeds back on the transfer from A to B, so that if C builds up, it switches off A to B and slows it down, the production of it. If, on the other hand, it goes down low, it speeds it up. It's a negative feedback. The opposite you see below it, which acts as a switch, is that when you make C, you promote A to B, and so once you've started it, it will turn into a switch, and you go into C. Now, this idea was first generated clearly by Jacob and Monod in the lac operon, looking at gene regulation, and they determined negative feedback. But a point is the same one as I've already said. You could describe the lac operon in terms of protein touching DNA, messenger RNA and the structure and so on, and you don't understand the biology at all. But once you've understood it forms a negative feedback loop and manages the information in that way, it makes complete biological sense. And so you're translating it into information, makes it sense. This sort of thinking leads to regulatory circuits which can form feedback control, switches, toggles, timers, oscillators, which are all combined together to form complex networks in living cells. So I'm now going to go into a couple of metaphors to illustrate how this might sort of be useful in thinking about biological problems. One is this metaphor. I dug this out of a journal, where the electronic circuit, in this case was, I think, a board from a radio, some wit has labeled the transistors and so on here with the names of genes important for cancer. And the idea here is, of course, that we should think about the circuits inside cells in the same way as one thinks about electronic circuits and the managing of information. I want to make one further point, though, is that this is a hardware circuit, and then we control it through software if this was a computer. Interestingly, living systems, the word wetware has been coined because you connect the different components through diffusible chemicals, and you can rewire the circuit by changing the way those chemicals work, or the elements within the wiring changes. So what that means is that you can actually deal with information not simply by programming, but actually by rewiring the hardware, which is why it's called wetware, which has sort of interesting concepts. So this idea leads you to consider the cell as a sort of complex system. It's acting as a logical computational machine of linked logical modules. And that's the way you can think about it. Second metaphor, transport hubs, complex systems. Here what you see is, of course, an airline map of flights going from different cities in the United States. Now, if you look at this, you would conclude what you have is transport hubs connected to places that are visited less often. And so you have New York and Chicago, or, for example, or Detroit, connecting to Dallas and so on in Texas. And what you can conclude by looking at this is that if you take out New York, the whole system collapses. If you take out Kansas City, nobody cares. Now, they may not care anyway about Kansas City. I quite understand that. That's right in the middle there. If I had a pointer, I could point it out to you. But this emerges from the structure of these maps. What's interesting is that in metabolism and in the behavior of the cell, there are similar sorts of hubs connected in similar sorts of ways. And why I bring this up is understanding complex networks at a completely different level, transport hubs or in social systems, is in fact very revealing about analogous complex networks which behave in cells. So there's something to be learnt about complex networks. Something else, networks and pathways. We tend to think, when we're thinking about biology, of linear pathways, arrows connected together. And that's quite simple to think of. You switch on the first arrow, you go down a series of reactions, you end up in the last arrow. This is the sort of thinking that many of us, or at least half of us, are very comfortable with in thinking. The reality isn't like that, for various reasons, partly due to evolution by natural selection, which leads to redundancies and so on, because you're always dealing with what existed before, is that complex networks have redundancies, they have connections, they're not efficiently designed, it's not intelligently designed. And so, whereas in that top pathway, if you switch off the first arrow, you will switch off the last. If you look at one of these arrows down here and you switch it off, it's difficult to predict what the outcome would be. And the reason I'm mentioning this is that biology is the bottom. And it's more sophisticated, it's more redundant. Occam's razor does not apply. That's very important, because when physicists get into this, they're always trying to apply Occam's razor. And you've got to be very, very reserved about this. And a second point is that we tend to think more in an easier way about linear pathways. We don't sort of want to construct the latter. I said half of us because I found this picture I'm a recreational pilot, and I found this in a pilot's manual. And men tend to think like the top, and they're either on or off. Women are more sophisticated and more nuanced, and it's more complex, and that's more like we see here. If I showed this in the United States, I'd get so much shit after the lecture, I have to say. But I'm hoping I can get away with it in Sheffield, okay? Another metaphor. You have pathways, regulatory connections. That, as you see from the bottom arrows there, could be, you'd switch on the first arrow, you get the second arrow. Now, that is limited information transfer. You know, it's on or off. But if you pulse information down that circuit, then you introduce dynamics into the system, and the ability of basic circuits to communicate more sophisticated information emerges. So, for example, on the top there, you see we're pulsing in a unit of time 4 peaks or 2 peaks, right? That can have a different meaning. And the point I'm trying to make here is introduce dynamics into such systems, and the ability to have more complex communication networks increases significantly. In fact, very significantly, and I'll go to another metaphor, which is from Morse. Morse code changes what is essentially a traffic light system, on and off, green or red, we'll forget about amber. But if you now introduce dynamics through dots and dashes, you can now write the works of Shakespeare. The amount of information that you can communicate if you have that sort of dynamics is enormously enriched. I'm utterly convinced that dynamics in biological systems are going to, which we've barely understood, we already have some evidence for it, not from my lab, from others, that dynamics is going to be very important. I am sure life has exploited this much more than we've had before, thought before. Generation of forms, spatial order, really interesting. Traditionally, the idea is that they haven't been very interesting. This is a bacteriophage, it has a nice shape, but it is all direct atomic and molecular interaction to make a shape. Interesting, but not really very fundamental. What you need is order over distance beyond molecular contact. You need fields to be generated, and those fields can be responsible for finding out where the middle of a cell is or where the middle of an embryo is, and you need that for spatial order. You can generate spatial order by other ways. This is a chemical reaction here, the Zhabotinsky reaction, which was modeled by Alan Turing, great mathematician from Bletchley in the 1950s, which gives you order across a structure. And almost finally, I need to illustrate another idea which is important in thinking about these things. What you see here are Harrison's clocks. Those of you who have read Darva-Sabel may know what I'm talking about here. This was a prize set up by the Admiralty, British Admiralty in the 18th century to get an accurate clock to measure longitude. So he designed the clock H1, top left, learnt from that, found it wasn't accurate enough, redesigned it, and you had the clock top right, same thing, redesigned it, and got 2 more clocks, ending up with H4 down the bottom there, completely different. Now, why I'm putting this up here is these clocks are intelligently designed. That is, you learn from the object, you then apply those principles more intelligently to get the next object. Life isn't like that. You have to evolve from one clock to the other. And if you're evolving from one clock to another, you have all the debris of the old clock, which is still with you. And this is the reason for the complexity, the redundancy, and all the things that doesn't apply, where you can't apply Occam's razor, because you've got all this mess. And it means that whilst we think we are efficient, what we really are is just more efficient, if you like, than our neighbours, and we are actually not very efficient at all. And we always need to be thinking about that when thinking about regulation and managing information, because it's not necessarily simple. Now, I'm going to finish with some last observations. Living things, as I've just described, are very complex, much more complex than the normal physical objects that, for example, physicists or chemists would actually study. And I wonder whether this complexity is going to lead to counterintuitive explanations. Because biological explanations up until now have more or less operated in what I call a common sense world. A common sense world of Kantian 3 dimensions of space and time, with sort of billiard balls in there knocking into other billiard balls. It's how we think about it, okay? It's how I think about problems. But when you have this level of complexity, I wonder whether we might move into a stranger world. And the analogy there is, of course, physics. Pre-1900, mostly, physics was rather straightforward and also occupied a common sense world. But post-1900, particularly when dealing with the very big and the very small, that is, problems at the level of the universe, problems at the level of the atom and below, physics became increasingly bizarre. And in my view, I wonder whether biology, complexity, may take us in the same way. Think, as I said, before 1900, physics sort of common sense. Then you have Einstein in 1905, and you have relativity. Now, there are many popular books on relativity. Now, what happens is you'll read a book on relativity, it'll have metaphors and explanations, you think you've got it, and you close the book, and somehow it just floats away, you know? And it floats away because unless you've really, really got it, particularly the maths and so on, it isn't part of our common sense world, this relativity and clocks going faster and slower and so on. It's just not the world we inhabit. It's not a world we feel comfortable with, so it slips away. And if you think you have trouble with Einstein, move on 20 years and you get into quantum physics, and then you're in pure Alice in Wonderland country. It is really impossible to comprehend it. And there's book after book on quantum mechanics and string theory and so on, and they're all equally incomprehensible when it comes to it. I used to think physicists understood it, actually. But when my daughter works at CERN, she's a high-energy physicist, and I realised that she doesn't get it either. And they have these constant seminars together to sort of encourage each other that somehow their world is making sense. And I think it's because they have to struggle with the ideas. I would struggle with it. It makes sense in the mathematics, but it isn't making entire sense in our common sense world. I wonder whether biology is on the edge of such a strange transition based on complexity and the fact that it is so unusual, so strange, that what it might produce. So these are my ideas. The cell, the gene, evolution by natural selection, life as chemistry, I think are given. Maybe information and systems as a way of explaining biological phenomena might be a fifth idea. Thank you very much.